Hello, everyone. My name is Abhya. I am a medical student from GW, here to talk with you all today about our lab's paper on computational analysis of cortical neuronal excitotoxicity in a large animal model of neonatal brain injury. So to start us off, we hit this key point that hypoxia causes neuronal dysfunction and death in the developing cerebral cortex, and that's due to excitotoxic calcium influx. What we have shown on the left in the image here is that you have a presynaptic neuron that is releasing glutamate. That glutamate can bind to your NMDA and your AMPA receptors. Calcium can flow in via those channels. That can also cause opening of these voltage-gated calcium channels, thereby resulting in additional calcium influx. So we see now that the NMDA receptor becomes of particular interest. It's regulated by several of the species that we ended up including in our model. I already mentioned glutamate to us, but additionally, inside of the postsynaptic neuron, we have got our SRC kinase, which I'm just going to call SRC kinase. We've got PKC, CAMK2, and we also have some regulation by ATP. So again, thinking about how hypoxia may drive this process as well. To bring this together with an in vivo model, we know that from our lab's previous work in translational piglet models of hypoxic encephalopathy, we've been able to show that hypoxia overactivates the calcium calmodulin signaling via SRC kinase in cortical neurons. And that's what you see here with this green calmodulin combining with calcium and that contributing to activation of SRC kinase. Ultimately, the upregulation of SRC kinase causes an increased transcription of pro-apoptotic genes like BAX. Using 2 to 3 day old piglets, use the cerebral cortex to quantify nuclear calcium and CAMKK2 expression. We also used quantification of ATP production to ensure that in the 3 conditions that we had these piglets in, we had actually achieved hypoxia. So those 3 conditions were a normoxia condition, a hypoxia condition in which after 1 hour under anesthesia, we decreased their FiO2 to 0.06 for an hour prior to harvesting the cerebral cortex. Of note, we did see then that the ATP was significantly reduced in this group compared to the normoxia group. And the third group was a hypoxia group that also received PP2, an inhibitor of SRC kinase. That PP2 was given 30 minutes prior to the hypoxia induction. But otherwise, the hypoxia and hypoxia plus PP2 groups had the same methods. What we found from this is that PP2 treatment reduced hypoxic injury-induced calcium influx. However, that calcium level in the hypoxia plus PP2 group was still higher than in the normoxia group. There was partial rescue there, but we did not achieve the pre-hypoxic insult levels of calcium. The other main finding that we had is using Western blot, we also characterized the CAMKK2 expression and saw that it was significantly increased in the hypoxia group as we expected. However, in contrast to what we saw with the calcium influx with just that partial rescue with CAMKK2 expression, there was almost complete respiration, almost to those normoxic levels that we had seen previously. This next set of figures is showing you how our model aligned with our experimental results. So you can see in A, in this figure, that there was an increased activation of SRC kinase. In B, you can see that nuclear CAMK4 was increased. In C, you can see that cytoplasmic and nuclear calcium were both increased. And lastly, in D, you can see that BAX expression was increased, all of these induced by hypoxia. So having confirmed and validated our model using our experimental data, we moved on to a global sensitivity analysis. We computed our Sobel indices to analyze how sensitive our model was to changes in concentration of different species. You'll see that each of these rows is focusing on STIM, hyperstim, NMDA receptor and nuclear CAMK4 as our inputs, varied those parameters from 0.25 to 1.75 times their starting concentration, and observed the nuclear calcium and BAX expression outputs from that. From this, we found that for BAX expression, STIM, hyperstim, and CREB all had first order non-zero indices, and the total order indices for these were higher than the corresponding first order indices, which you can see. We then moved on to a multi-parametric global sensitivity analysis, and the goal there was really just to identify the specific conditions where the input parameters of global sensitivity analysis had differential effects on calcium influx and BAX expression. So we plotted all of those simulations from the GSA, and 2 of them showed a dissociation between BAX expression and calcium influx after giving PP2. You can see that on the dotted lines with the asterisks in the top and bottom panels here. And so since there was a threshold then above which our simulations had this calcium influx and BAX dissociation, we created a classifier for the multi-parametric global sensitivity analysis based on that threshold, where we set the maximum values per simulation of 10 micromolar for nuclear calcium influx and 1 micromolar for BAX expression. Then we reuse the same Sobel indices that we used for the global, or that we calculated for the global sensitivity analysis for this multi-parametric global sensitivity analysis to come up with empirical cumulative distribution functions to accept or reject our simulations. Ultimately, we found for Bax expression that there were significant differences for hyperstim and for Krebs. Altogether, the multi-parametric global sensitivity analysis just further confirmed for us that Krebs and excitotoxic glutamate pulses significantly affected Bax expression, and only excitotoxic glutamate pulses could affect the calcium influx. We also did parameter scans where we altered the reaction kinetics of the NMDA receptor. So our results here show us that above a Ka of 5 times 10 to the negative fourth, Bax expression drops off significantly during PP2 administration, but the drop in calcium influx actually is a little bit delayed. So we get a significant reduction for calcium once we get a Ka above 7 times 10 to the negative fourth. That suggested to us that generally altering NMDA receptor activation will only cause calcium influx Bax dissociation in a limited range. That also suggests to us that there is some discontinuity in NMDA receptor signaling that results in the activation of these cell death pathways. By contrast, when we altered the reaction kinetics of MK2 activation, we saw a uniform relationship between calcium influx and Bax expression during hypoxia. But for Ka values that were less than 9 times 10 to the negative 4, we saw that calcium influx. Last graph that I wanted to share with you really hits on something that I think is clinically relevant. Many of the SRC kinase inhibition studies using small molecules like PP2 have focused on pretreatment with PP2 prior to the hypoxic insult to see if that prevents the kinds of changes that we see after hypoxia. However, to make this clinically applicable, we have to consider if that same rescue is actually occurring when we treat after the hypoxic insult occurs, as would happen in practice. And so these graphs show you essentially that if you compare PP2 pretreatment simulations with simulations where PP2 administration followed hypoxia, and if you can combine that with alteration of that interaction between SRC kinase and NMDA receptors, if you can change how strong that relationship is, then pretreatment you can see clearly prevents that significant calcium influx and prevents that increased Bax expression. If you also increase the Ka of SRC activation of the NMDARs by just 20%, that will also give you a large drop in cytoplasmic calcium, and you'll get Bax expression that returns almost to pre-treatment levels. So that suggests to us that PP2 administration that follows hypoxia could still be beneficial, and clinically that could still be relevant, but that we could dramatically improve that effect by focusing on incorporating other small molecules that specifically target that SRC and NMDA receptor interaction. So I'm really hopeful that using this computational model, we have been able to gain greater insights into what key features to kind of target the pharmacological interventions as we work toward preventing the consequences of hypoxic injury to the neonatal brain.